Hello, everybody. Welcome. I'm going to start this webinar, so we're on time. Welcome to an Immune Effector Cell Standards, our second edition webinar. Glad to be here to introduce our speaker. My name is Dr. Kevin Curran. I'm a pediatric bone marrow transplant and cell therapy doctor at MSK. But our speaker for today is Dr. Nic, who is the FACT Standards Committee Chair and the Cell Therapy Lab Director at the Alberta Blood and Marrow Transplant Program. You can use the chat box down the bottom to ask questions anytime during the session. And after Dr. Nic's talk, we'll have a chance to go through those questions and hopefully be able to clarify any updates to the IEC standards. Quick bio by Dr. Nic. Dr. Nic is currently the Cell Therapy Lab Director at the Alberta Blood and Marrow Transplant Program. In this capacity, for both adult and pediatric patients, she serves Foothill Medical Center, Alberta Children's Hospital, the Tom Baker Cancer Center, and the Calgary Lab Services. Dr. Nic obtained her PhD in biochemistry from the University of Saskatchewan. After completing her studies, she did 10 years working in Houston and Philadelphia, where she did research on gene editing technologies for blood stem cells for hemoglobinopathies. She returned to Canada to assume this director role at Alberta. Throughout her career, she's written numerous articles and presented her research findings in an international capacity. She currently serves as the FACT Standards, the chair of the FACT Standards Development Committee. She contributes to the development and enhancement of quality standards in the field, and her life's dedication and research is to improve patient outcomes. She's also been a FACT inspector since 2013. So with that, I'm going to turn it over to Dr. Nic.
Good morning, everyone. Thank you very much for attending today. Today's topic is, as you all know, the Standards for Immune Effector Cells, second edition. As mentioned in my bio, thank you very much, Kevin. I'm the standards chair for FACT, an inspector, and in my day job, I work as the director of the Cellular Therapy Lab here in Calgary, Alberta, which is an adult pediatric program actively involved in both transplant as well as cellular therapy. So what I'm going to do today is kind of give you some of the just a note on disclosures here, nothing really related to what we're going to be talking about today, but I like to present those there. The objectives today for our talk are basically to provide an understanding of the intent of the Standards for Immune Effector Cells, IECs, and kind of what they relate to, how the standards are developed, revised, as this is a new edition, and the importance of things like public comment in those to make sure that we can bring forward the best standards for immune effector cells. And I really want to highlight today some of the key changes. There's been a lot of changes made between versions 1.1 and 2.0, so I want to highlight some of those key changes. This is not going to be a complete and comprehensive list of all the changes. That would just take by far too much time. They were published in December 2023. So the Standards for Immune Effector Cells, this is the second edition, version 2.0, which was, as I mentioned, published in December of 2023. You can find them, of course, on the FACT website, factglobal.org, as well as you can purchase hard copies and download copies of both the standards as well as the accreditation manual there. The second edition for the FACT Standards for Immune Effector Cells is effective March 15th, 2024. So that's why we wanted to bring this presentation here today. As well, there are crosswalks available and other helpful documents to help you navigate through the different versions to make sure that things are quite clear. So just a little bit of background about how the standards are developed. This is kind of, as being the FACT standards show, this is kind of my first go around with going through the whole process here. So I've learned a lot, and I think it's really important that everybody understand that these FACT standards are developed by consensus of experts active in the field and are evidence-based whenever possible. Where there are very few definitive studies or publications, especially with what a lot of these cutting edge technologies, the Standards Committee weighs the available evidence from the preclinical studies and utilizes accepted scientific principles to reach consensus on what we should have for standards. So the standards are available for public and regulatory comment prior to ever publication. And each comment is reviewed, and the standards group committees review them, respond to them, and revise if appropriate on those comments. So really the comments of everybody working in the field is really important to make sure we can make these as robust and as beneficial as possible. So the FACT standards for immune effector cells were developed by consensus of experts, as I mentioned, working in the field of laboratory practice and in clinical practice related to immune effector cell therapy. The immune effector cells are defined as cells, in vitro modified or not, that have differentiated into a form capable of modulating or affecting a specific immune response, including T cells, tumors infiltrating lymphocytes, TILs, natural killer cells, dendritic cells, and mesenchymal stromal cells. So of course, as we all know, the CAR T cells fall under this category as an example. The scope really is the care of the recipients across that whole continuum of care, from diagnosis through various therapies in the inpatient, outpatient, ambulatory settings for a lot of these now, short and long-term follow-up, management of complications, adverse reactions, and importantly, outcomes. And reporting. The donor is also needs to be followed throughout the selection, donation, and follow-up, whether that's an allogeneic or an autologous donor. So the scope of these standards, as I mentioned, goes across all of these phases: collection, manufacturing, transportation, shipping, administration, during all the phases of both clinical trials as well as the standard of care product. As we know, it's very, very important to make sure we maintain things like chain of custody and chain of identity in these. The cellular therapy programs across all facilities for clinical care, cell collection, cell therapy processing, and manufacturing are included in this. Important to note in the second edition is the scope has been expanded to include collection of cells from cadaveric donors as appropriate. So we now have terms living donors. It was changed to donors, for example, to include that. So these standards are intended to be flexible. All of the standards are, because there are a lot of different models of patient care when they're using cellular therapy products, as I mentioned, inpatient, outpatient, ambulatory care, etc., as well as collection at very different locations. So these standards include all the requirements for programs administering immune effector cell therapies, but not hematopoietic cell transplant. So these are essentially what we can refer to often as standalone programs. Programs that administer IECs within BMT programs, the standards for those programs and the administration of the IECs within a blood and marrow transplant program are incorporated into the FACT-JC International Standards for Hematopoietic Cellular Therapy Product Collection, Processing, Administration, which is version 8.1 currently. So feel free to look at that as well too on the website. So let's get really into the nitty gritty of everything today and talk about the key changes that we are seeing in the IEC standards version 2 or edition 2, the second edition. And a lot of these are to align with the HCT standards version 8.1 to create some sort of harmony across the board and start to work to have these standards harmonized as best as possible. So one of the first updates was actually the addition of chain of identity and chain of custody. So these terms were defined by a multi-stakeholder chain of identity and custody working group and the standards coordinating body. And these terms have been added. The concepts have always been there of, identifying and your donor and your product recipient throughout, and ensuring we know exactly where it's been. But really, these chain of custody and identity are there to permit necessary critical tracking and tracing of products. So the actual definitions are in A4, and basically chain of identity is making sure you can identify that product through its full life cycle. And then we also have, we need to make sure that it's permanent, readable, and present for all of those things that we need to have available. There are several applicable standards on these in all of the sections B, C, and D. So really a lot of the key things is ensuring that, when you're collecting the products or preparing, that, the processing is done at qualified sites for collections. They need to have chain of identity in place. There's portions of the quality management plan that, you want to make sure that you have a very clear definition and process and standard operating procedures in place to ensure both chain of identity and custody allow for tracking from donor to the recipient to the final disposition of that product. And then, of course, with that, it's really important to do annual auditing at a minimum to ensure that the appropriate chain of identity and custody is being followed with those products. The second concept that I want to bring forward is an alignment as well with the HCC standards, is the genetic modified cells. And basically, it's a term to define, what these cells are and how they're used. So the definition is a cell that has been modified by genetic transfer or editing for therapeutic intent. And of course, the use of these cells is very important in our immune effector cell world, but it also requires a lot of additional review and outcome analysis, biosafety planning, etc., and important regulatory requirements for these different modified products. So, of course, you will see standards throughout the entire IEC, standards relating to genetically modified cells and what needs to be done. Additional training for physicians and nurses, of course, important for outcome analysis, safety, and efficacy for these genetically modified products, and as well, really important in the processing to ensure there are defined areas that are present to prevent mix-up, appropriate labeling, contamination, cross-contamination of these cellular therapy products or genetically modified products. Additionally, of course, a big thing with the genetically modified products is the need to have biosafety in place. So there needs to be biosafety plans that are appropriate and in accordance with your institutional biological safety committees, and that are comply with the applicable laws that are present in your jurisdiction. So in addition to that, of course, disposal becomes very important, whether it's disposal, on the clinical side or in the processing facility side, to make sure that those meet the appropriate biosafety levels as well as the waste management levels based on your laws in your jurisdiction. The third alignment that was done was the introduction of the term GXP. And this is defined as good practice with the X being variable. And this was further defined good practices based on different applicable laws and industry standards and the type of work that is being performed, what would actually apply. So examples of this would be good manufacturing practices, GMP, good documentation practices, GDP, good laboratory practices, GLP, etc. So these standards also require annual training in those applicable good practices that will be based on what the requirements are for the work that you've been doing and what your program is involved with. So that's the definition that is currently in the standards. Good practice following various quality standards and regulations. The X is variable with further definition of good practices defined by different applicable laws and industry standards. It is based on the type of work being performed, which the GXP should be following. So here are just some of the examples of the standards that highlight the need to have training associated with GXP as appropriate. So the training, of course, is in all of the parts, B, C, and D, annual training and applicable processings performed in accordance with applicable law. In the guidance here, we have some examples. Basically, training must be provided and documented at least annually for all relevant employees. The exact content needs to be determined by the program. So you have to have some training policies, procedures in place to clearly indicate what kind of training needs to be done. The number of hours is currently not specified. It is specified for things like standards, CME, and etc. But the number of hours specifically for this type of training is not defined. The program needs to determine what is appropriate. The fourth area is again on the importance of addressing risk management standards in programs that are using these products, these cellular therapy products. So general standards were added to address risk management program requirements for clinical programs utilizing licensed or equivalently regulatory approval cellular therapy products for which a program is required by applicable law or the manufacturer to have in place. So it's really important that the clinical programs establish and follow policies, SOPs, related to any mandated risk management programs that are needed. And that includes both training and competency of staff on that risk management program. So to make sure that they have, for example, medications to deal with major severe adverse events with specific products, reporting of those adverse events, things like wallet cards or other means of communicating with the recipients and caregivers and other healthcare professionals who may need to know and are providing care to that patient. It's really to ensure that we can deal with any adverse events that occur in these patients and make sure that there's consistency and continuity of care. So a risk management plan is expected with this new version of the standards. The definition, a document that describes the current knowledge about the safety and efficacy of a cellular therapy product and the measures to be taken to identify, monitor, prevent, or minimize risk associated with the use of that product. So as you can imagine, this could be quite specific for different products or categories of products that fall under the IEC definition and scope. So here are some applicable standards in the clinical program right here, B710, where it needs to be in place, and it spells out things like training and competency, management of adverse effects, necessary medications in place, reporting them, as well as communicating with wallet cards or other means of communication to make sure that the entire care team has this information for these patients. So I'm going to go on now to talk about actually one of the biggest changes that you'll notice in the standards right now, and that is to the standards surrounding the quality management standards. And of course, this is, again, something that I can't go into every single detail of the changes that occurred, but I just wanted to highlight some really important factors and things that have come up and concepts that we want everybody to be aware of. Of course, I've already mentioned annual training for things like GXP and having it in the quality management plan, at least a summary or reference to the policies and standard operating procedures for that. Additionally, things were added to change the requirements for the document control system, things like including critical document types that needed to be controlled, the review every 2 years at a minimum, and having things like document versions available and communication of training of all these things. So good quality management processes. Also, we want to have things in the quality management plan that speak to safety and efficacy of the cellular therapy products that you're utilizing. And importantly, of course, reporting and doing outcome analysis to verify what you're doing is actually providing that safe and effective product. So, of course, some of the audit requirements have been revised, and those are across all of the standard parts. B, C, and D out of 4.8. So As of note, it's very important, of course, to have additional standards have been placed for related to products with positive microbial testing. And this has actually been used to harmonize with all the sections in the standards, because it's very often that when a microbial positive result comes up, it's collection, clinical, processing all have to be involved. And so everybody has to have a policy and procedure in place to make sure that that's handled. The quality management plan has also been further enhanced in regards to occurrences, and this has been harmonized across the sections as well, including 2 additional requirements. So one of them is basically to determine that a cellular therapy product has resulted in adverse event or reaction, that basically it's both if there's a donor and recipient's physician should be informed of this, to make sure that the donor is aware of this and can be checked for any adverse effects. Occurrences also need to be tracked and trended as part of a good documentation practice for this. And that's, of course, across all sections. So really, yes, again, there's been addition of adding in requirements for qualification of things across the clinical section, and this is to harmonize with collection as well as processing. As well, there needs to be information about things like verification and validation of critical procedures in both the in the clinical side, in addition to collection as well as processing. That has been included in the new version of the standards. It's really important in the standards now to have some policies relating to management of clinical supplies, including drugs and supplies. We've seen a lot of issues, as everybody knows, with shortages in various supplies, from various drugs to tubing sets, etc. So it's really important to have appropriate procedures in all of your sections for how you deal with this, and if you have backups of supplies, etc., and how those have been looked at, to ensure that you can continue functioning when there is shortages. The quality management plan should also include, again, summary and reference to policies about evaluation of risk and change of process. So again, certainly the pandemic and other things raised a real sight to be aware of, that, processes may have to change, and how do we evaluate the risk if there's some sort of critical event that occurs, and how do we critically change those processes. And that's on the clinical side in 416, and also has been harmonized with what is currently there for collections as well as for processing. It's really important that your clinical program director, collection facility medical director, processing facility director are involved actively in reviewing the quality management plan and the activities associated with that. The activities at a minimum, there are certain elements that you want to look at quarterly, and then yearly you want to do a review of the effectiveness of your quality management plan to ensure that it is meeting all of the needs of your program. There's also been an additions in the clinical section on clarification of some of the emergency and support services for IEC recipients, for needs in intensive care units and emergency services, additional supports that they may require by trained personnel, the need for things like irradiated blood products, CNV appropriate irradiated blood products, and as well, there needs to be qualification of the sites that are actually doing those collections. So I just also here, another one is external party agreements. So this is in the clinical section. I just want to highlight that, there should include the responsibilities of the external parties to provide clinically related information related to products and services has been added. But really, there's a whole section that I encourage everyone to look at for external party agreements, making sure you have those appropriate agreements in place with any external parties that are involved in providing you supports or services or products. Specifically in the collection standards, as we mentioned right at the very beginning, the scope has been expanded to include non-living donors, so that's why it's been converted over to donors. So that's one of the major significant changes to meet those needs. Also, there's been inclusion of chain of identity concept during collection. So there needs to be processes to qualify the site for cellular collections, and that needs to include the ability to have a chain of identity. And of course, that also includes surgical collection of cellular material. We need to make sure that there's that chain of identity is included in any of those products that are going to be used. Personal requirement is unchanged. The accreditation requirement is new. So the cellular therapy services shall be overseen by a designated medical director, quality management, and a minimum of one additional designated staff member. This team shall have been in place and performing cellular therapy product collections for at least 12 months preceding initial accreditation. So that's C14. So to show that there's some history of experience there before initial accreditation is done. And then there needs to be a minimum of 5 cellular therapy products collected during the 12 months preceding initial accreditation, and a minimum average of 5 products shall be collected annually within the accreditation cycle. So that's C1.6. Some additional standards changes include a subdivision of collection standards based on IEC source, just to kind of delineate and show that there is a separation and often different requirements for some of these products. So C 9.1 is collection of whole blood. 9.2 collection of apheresis products. So those all kind of fall under peripheral blood collection. C10 is marrow, and then the collection of other tissues. And of course, there are additional standards now for product storage, transportation, and shipping, C12 and C13, based on what products are stored, what products are collected by that program, and whether they're transported out of the facility or shipped out of the facility. All of that information is included in those standards and the requirement to have appropriate SOPs for such information. So the processing facility, one of the first noted things is expanded requirements for collection kits. Often these are provided by the processing facilities to whatever collection site. So D211 speaks to when a collection kit is prepared and sent to a collection staff, there needs to be adequate instructions and materials to collect, label, store, pack, and transport or ship the cellular therapy product and associated samples to the processing facility. So there just needs to be good management of SOPs and documentation to ensure that all of that information is available. Some additional ones would be, there's some expansions on requirements in training and continuing education within the processing facilities, because of course, we are dealing with a variety of products that are present there. So it's important to have appropriate training and continuing education on the products that you are working with and processing and involved with, as well as the quality aspects of such things. So as highlighted in D 4.4.2.4, continued competency for each critical function performed, assessed annually at a minimum. So ensuring that your staff is competent on any of these critical functions that are being performed, especially important if procedures are only being done rarely, or you have a large number of procedures. You want to make sure that your staff is competent on all of those critical functions. Annual training, of course, in appropriate GXP to the process being performed in accordance with applicable law. And then, of course, inclusion of outcome analysis is very important in all of the sections, and it's highlighted as well here too in the processing section to include in the quality management plan. A review of outcome analysis and product efficacy should be included. Minimal endpoint of clinical function as approved by the clinical program director, because it may be very different for the different types of products we're dealing with. Also, there needs to be review in the treatment-related morbidity morbidity and mortality at 30 days, 100 days, and 1 year after the cellular therapy product is administered, or in accordance with applicable laws. Data outcome analysis and cellular therapy product efficacy, including any adverse event related to the donor, recipient, or product. Those shall be collected and made available in a timely manner to clinical collection or distribution personnel as applicable. So it's really important to work with all of the other components to make sure that you're getting that information out there so it can be analyzed appropriately with people. Audits, of course, with the addition of the terminology chain of identity and chain of custody, it's very important to do audits on those to ensure that that is being used appropriately and there is no breaches in identity or custody of these products. There also needs to be policies or standard operating procedures for the management of any external audits that might be required by commercial manufacturers or applicable regulatory agencies related to these products. So I think that was a quick and whirling discussion of everything, but I wanted to make sure I left time open for questions today, as there are a lot of new changes. The key takeaways that I've put in today from this are kind of that I want everybody to be aware that, yes, there's the FACT standards for Immune Effector Cells second edition was published in December 2023. I want to say a thank you to everybody that was involved in the work of this, all of the volunteers that were involved in the committees that worked towards this, as well as all of the FACT staff that spend tireless hours to make sure these things get put out and are as best as possibly can be. And they will take effect March 15th, 2024. Some of the big highlights that I put in the discussion, and we're happy to go over in detail in questions if anybody has them, is the concept of chain of custody and chain of identity. These are not new concepts in a lot of senses, but really ensuring that these are in place for all of these products from the start of working up your donor to the final use or disposal, and then outcome analysis. You want to make sure that information there. The importance of GXP training, and that's appropriate, good practice training in the areas and the levels of processing or collection that you are providing of these products. Of course, risk management plans are very important, and there's a requirement for them. And this is highly dependent on the products that you're doing, the requirements of your laws, as well as the external agencies or corporations that you're working with. And of course, the biggest change I would say that we see overall here is that expanded quality management plan requirements. And this is really to harmonize with the other standards that are out there and ensure that we are providing the safest and best products for our patients that are receiving these immune effector cells. So with that, I'll just put up on the screen some references that are available here. Of course, please go to the FACT website where you can get links to all of these standards pages as well too, and to the store for downloads or purchase of printed copies. I think it's really important to review those. And of course, you know, it's important for you to look at not only the standards, but also any of the guidance that's associated with that. And with that, I will end my talk, and I think Kevin's going to come back on now.
Yes, thank you. Thank you, Nic. That was fantastic, and thank you for that. We're going to go over some questions. Before I do that, I do want to share the location and recording of this talk. So there were a number of questions on getting these slides, and yes, you can have access. You can find the recording in PDF of the slides within the FACT portal. If you visit www.factglobal.org and log in, and you can find under meeting documents, and they will be the updated final version that you're seeing today. There were a number of questions because the ones there were the draft versions. Okay, so we have a number of questions. Bear with me. We have a number of questions that have come out. And so, Nic, I will ask you the question, and between us and potentially Phyllis, our Chief Medical Officer, may join us.
Oh, excellent. There's
There, so
Phyllis, I
two
noticed
for
she's
Paul.
already been answering some in the chat, so that's awesome.
So the first question comes: does DLI fall under IEC? So I can answer that. It does not. So donor lymphocyte infusions, because it's from the donor, that is tied to the transplant. So DLI is under the transplant standards.
Right.
The second question from Cassie: is there an ETA on the self-assessment spreadsheet for version 2? And the goal is for that to actually be posted next Monday. So Phyllis was able to get that information. But so be on the lookout for that very soon. We had a number of questions about when the full second edition IEC manual will be available. People really like the explanation and evidence that goes with that. The goal for that and the target date is mid-March. You can now look in the transplant 8.1, so there will be there's a lot of overlap. It's for those who are just focused on IEC and maybe don't have a lot of overlap with the transplant. There's actually a fair amount of overlap in the standards. And if you use the transplant explanations, you can actually find a lot of the key information. But we will have that out by mid-March. Here's a good one for you, Nick. Could the speaker touch on the components of a biosafety plan?
So the first thing I would say in regards to this is it's really important to work with your biosafety group at your facility to see what their requirements are. They are the experts on the needs for what you need to have in place. Usually in these plans, you have to have in place appropriate training, appropriate information for staff on the products that you're currently using, appropriate disposal, appropriate emergency and safety responses, any reporting that needs to be done to federal agencies. Being from Canada, we have to report things to the Public Health Agency of Canada. In the States and in Europe, it's different depending on who you're interacting with, on who needs to report to it. So the reporting is very important for this. So all of those things need to come together in your biosafety plan. Most larger institutions that are already dealing with some biological products already have one in place. But the important thing is to ensure that that is tailored appropriately to your cellular therapy products, because of course it's going to be quite different for a cellular therapy product or an immune effector versus using, say, an antibody or some other biologic that might be in place or meet your needs. It's also really important in your biosafety plans to ensure that you have an appropriate understanding of the regulations for your current jurisdiction. So those would be my first immediate things. I don't know, Phyllis, if you'd like to add anything else in there from the US
No,
perspective on that for everybody.
I think that was nice. Thanks.
Perfect.
There's a couple of questions, Nick, on the disposal. So one came in. What are the common differences in disposal of genetically modified cells as compared to regular biohazard and universal precautions?
In a lot of places, I would say there aren't that many differences, but usually you have to have appropriate documentation of the disposal. So, just a standard biohazard, you often have a biohazard bucket. For some of the genetically modified cells, they want to actually have a defined, this is the location it came in. It was decontaminated, the outside of the bucket before it came into, before it was or after it was sealed up, marked that it did have a biohazard on it, what the biohazard rating and the might be for it if it's significantly higher for certain viruses, for example. And that's part of your biosafety plan. Is your biosafety group will see in Canada, we have different ratings of 1 to 4, basically, of what the level of biosafety concern is. So that information will say you must dispose of it in that way. But there needs to be better documentation for a lot of that.
Related sorry, go ahead, Phyllis. Were you gonna?
No, go ahead.
Related, there was a question from Michelle Gregory. Is there anything different for disposal of stem cells?
Hi, Michelle. Good to hear from you. No, there's not actually. In most cases, I would say, I think, a lot of these we contract out to companies that are designed to dispose of our biohazards at varying levels, and it's just important to document that we work with them, and these are what we're disposing of, and this is how they're being disposed of appropriately.
Okay, switching gears, there's a question about standard B4425. Can you please clarify what FACT expects of IEC clinical programs for annual GXP training? MDs, APPs, RNs all go through formal GCP training every 3 years, but unclear what sort of assessments we need for additional GXP training and how FACT would expect to see that implemented and documented.
I'm gonna pass that to Phyllis first.
Okay, well, I can take a start at this. I think at a minimum, you could consider something like good documentation practices in the clinical setting, because some of those requirements, some of the clinical documentation is still on paper, and so those kinds of signatures. Many times forms are handwritten and things need to be legible. You have to have identification of who put the entry, date and time, and proper methodology for making changes in documentation. Also, there's all the standards around electronic records, and the not that you are in charge of the electronic medical record, but there are some principles that you should be aware of and be trained on, such as having your individual access, having people have double check entries before they're actually entered, and there are some standards around cut and paste in the electronic medical record. How do you manage that, and how do you ensure that you're getting the right information into the record? And so people need to be trained on all these things. I think it's probably a little bit of GXP clinical has to do with when you are handling products that are licensed products, there will be REMS requirements around those. And those are some sort of good practice. It's not GMP. It's not that we expect you to be able to run good manufacturing practice compliant laboratory, but it's more these things. And that's why the term as applicable is in the standard.
Thanks, Phyllis. Related to that question, which I noted, and so does that mean the form of GCP training is an annual endeavor? I know at my own institution it's every 3 years also for the GCP training.
Well, so I think good clinical practice is more, I think that's more about running clinical trials. And so we haven't said that has to be annual, just because if it's a requirement that it's every 3 years, I mean, certainly on the year that you do that, that may be part of your GXP training. And then the next year you do something different. So we don't say how many hours it has to be. We don't say what the content has to be. And it doesn't have to be the same every year. And it definitely needs to be tailored to the activities of the program and cover the things that you do. And if you don't do certain things, then that's not applicable.
Thanks. All right, a question from Lorraine Greathouse. We are currently a transplant cell therapy program with FACT accreditation. BMT physician on our org chart is also part of the regenerative medicine team and will begin treating solid tumor patients with commercial TILs in the coming weeks. Products will pass through non-FACT accredited labs, but infusion will occur in the BMT inpatient by our trained BMT team. Commercial TILs manufacturer does not require FACT accreditation, but BMT resources are being used. Do the FACT standards, so here's the question. Do the FACT standards apply throughout the entire process? Patient selection performed by a non-BMT physician and team. Storage of the product in a non-FACT accredited lab. Confused by the scope of FACT in this manner.
So this is a challenge. And I think the main concept that we've tried to use is that the physician caring for the patient determines kind of where the FACT standards start and stop. And we certainly would say we're not requiring that TILs be part of your BMT program. If the patient is being treated on the BMT unit by the BMT physicians, then it is part of the program. If they're being treated by someone else on a different unit, then you can decide whether that's going to be part of your accreditation or not. It works different in different institutions. I am concerned about the concept of it passing through a non-accredited laboratory, and would wonder kind of why that would be for a commercial product. If what we have been classifying these quote unquote non accredited laboratories might be a spot for short-term storage of a commercial product. Having been manufactured, it is shipped and it has to be stored somewhere before it can be administered. And that somewhere still has some responsibilities. And what we will be seeing in the next edition, clarified a little bit better, will be that whoever orders that product. So if it's the clinical physician, that team needs to be responsible for where the product is stored short term when it gets to your facility. So if it's a not accredited lab, you still have to ensure that place is secure, it's appropriately clean, it has appropriate monitoring, and that the temperature of the product that's being stored is maintained appropriately and documented. And there's good chain of identity, chain of custody from the time that the product arrives at your institution to wherever it stays short term until it gets to the patient, and then that there's good documentation. It's the right patient, right time, right drug. So while you might have a laboratory that is not accredited formally for anything else it does, it's still up to the clinical program that's taking care of this patient and this product to ensure that short-term storage place is sufficient by meeting the standards that apply to short-term storage. And so I think in the next year, one of the challenges that FACT will have is to provide some examples of how programs can be organized effectively, and what some of the options are for who's in charge here, and when do the standards apply and when do they not. For the moment, we have already published in our HCT standards some examples of how one example, in one case, the solid tumor team may be just renting space on the bone marrow unit, using that room because it's a clean room, and there's good environmental monitoring around that space. But the physicians are not part of the BMT team. They may rent the nurses also, but it's not part of the integral care of that patient by the BMT program. Then that can be really outside of your BMT accreditation. So there's lots of ways to organize it, but I think the key piece would be the clinical team managing the patient determines whether it's part of your accreditation or not.
Thank you. Back to the questions. This is a quick one. Would TOSI be considered a critical supply? And the answer is yes. TOSI is a clinical critical supply. Okay, this is related to standard B4838. Could you please describe an example related to that standard? Annual audits of infectious disease resulting from a cell therapy product collection, processing, or administration. And how is this different from the standard around management of cellular therapy products with positive microbial culture results? How would FACT expect this audit to be designed and conducted?
So there might be some overlap, right?
Yeah.
With the positive microbial cultures, but there's other diseases that could be transmitted from a product, such as hepatitis, HIV, CMV, EBV, any number of things that we normally would test in the donor prior to the donation. But there are situations where some positive infectious disease marker tests are positive in the donor, and that product is still used. So what we would look at really is if we would expect the program to have an audit of their patients for those kinds of things, both viral, any infectious disease that occurs. Ask the question, is it possible this was related to the product? Was it a viral thing? Was it a positive microbial bacterial culture, a fungus? So there is overlap, but it's a little bit broader than that, and it's not from the perspective of the product, but it's from the perspective of the patient.
Okay.
Yeah, and I would say it's also important in these audits as well too, to make sure, you are looking to make sure the management was done in an appropriate way based on what your SOPs, your policies and procedures are stating how you should follow up on, say, a positive microbial culture in a related or an autologous setting or an unrelated setting. So it's important in those audits to make sure you are actually doing what you said you were going to do, and it results in the appropriate result.
Okay, we have 10 minutes left, but we still have a lot of questions, so we're going to
Okay.
keep going, but the hour is drawing near. So a question, are these standards applicable to standalone programs? And the answer is yes, these standards are for standalone IEC programs. Question from Patty Korth. Is it required to label all of these products with I think she missed the ISBT labels, even if the pharmaceutical company provides a product label?
So are you talking about the finished product, final product label? We don't require that licensed products with an approved finished product label be relabeled prior to administration. ISBT 128 would come into play at the time of collection of the starting material, and in any products that the institution manufactures itself for clinical trials in that institution or in multi-institutions. Also be aware of the new hybrid or split label that's available through ISBT 128, where companies are starting to use that at the beginning of the collection so that they can use ISBT labeling from the time the starting material is collected through manufacturing, and then include those ISBT 128 identifiers when the product comes back. But relabeling is not required if that was what the question.
Okay, a general question. Does TILs, genetically modified cord blood, and gene edited product fall under IEC?
Yes. Well, so you take one step back. Some genetically modified products may not be IECs. They may be transplant grafts, such as some of the genetically modified cells for sickle cell or thalassemia, those kinds of things. Those aren't really IECs. Those are transplants.
I know in my own program, I think of cells if they're going to differentiate or progenitors, they don't fall under IECs. Of course, if the progenitor is a T cell and it goes on to make more T cells, then yes, they would. So
yeah, right.
You have to think of the biological origin of the cell. Okay. So risk analysis need for all IECs that we plan to infuse to our patients, regardless if commercial or research product. The answer is yes. Can you discuss the fact requirements and scope, the standards regarding AE reporting for commercial IEC products?
Well, I think a lot of that is going to fall under what the adverse event reporting is as per the commercial IEC product, and the quality agreements that have been established between the infusion center, the clinical program, and that commercial group, as well as what the expectations of your regulatory law or applicable law requires you to report on those. So it'll be
and what the company has decided they're
what the
going
company
to
has
do.
said. Yeah.
If they are going to use reporting through the CIBMTR and its cell therapy division, then you do that. And if they aren't, then you can report according to manufacturer's requirement.
Back to I think these look like pretty new. I just want to go. Patty Korth is writing back about the ISBT labeling. It was for collection and processing, and I think we answered that. Yeah, you need to have proper labels for it, but we're not asking you to relabel the final product.
Right.
There is a question from Miss Ramsey. Could you please clarify fact standing on bispecifics?
So we've said bispecifics are outside the scope of standards, even though there may be some toxicities that are similar to those you see after immune effector cells. Bispecifics are not cells, so they're not within scope.
And I think that we've pretty much got all the questions. There's a very open-ended one. Is there, where do we need to beef up the standards? But I think we would need another hour to do that. I also realize
Well,
I
so we
realize
should say we are working on the 3rd edition of this
Exactly.
status standards, and they will be out this summer for public comment. So please do look at them. And if you think there are things that are missing, please comment. You may find some new things in that 3rd edition that are important and may remind you that there are other things that you want to add. So please do look for those standards to be out for public comment and submit your comments.
Okay, I do realize when I was sharing, I didn't share my screen last time when I was telling you where to get this, and there were a number of comments, so I wanted to make sure I did that before we closed. But remember, the recording and PDF of today you can find on the FACT website, and this is what it'll look like, and look for the meeting documents tab. I think with that, I think we can close, and I appreciate everyone's attention. It was a very well-attended seminar, and thanks for everyone. I don't know if there's any closing comments, Dr. Nic, Phyllis?
Thank you very much, everyone. We're really looking forward to continuing these standards and improving on them with the next edition as they go along.
And thanks to you, Nic, for preparing and presenting this webinar, and Kevin for hosting. We appreciate everybody's participation, also all the attendees.
Thank you, everyone. Have a great day.